Continuing on with microscopic anatomy of your skeletal muscle, we need to look at the individual components inside each cell. Take a little further look into what they are and what they do. So first off, the term muscle cell and muscle fiber. They mean the same thing. They're used interchangeably back and forth. The term sarcolemma. Now the sarcolemma is the cell membrane. Just it's a special name because now we're in the skeletal muscle cells. So sarcolemma. This would be covering the entire outside of the actual cell. We then have the sarcoplasm. Basically, it's cytoplasm with a slight modification to be inside the muscle cells. Now, this sarcoplasm, just like cytoplasm, would fill in the spaces inside the cell. So your sarcoplasm is your cytoplasm, but inside the skeletal muscle. Sarcoplasmic reticulum. Now, sarcoplasmic reticulum is pretty much the same as the smooth ER, or the smooth endoplasmic reticulum. You can see here, it's all this yellow that's being drawn around. It's going to wrap around and cover around the individual myofibrils inside the muscle fiber. T-tubules. Now they're also known as transverse tubules, but T-tubules is easier. These T-tubules are interesting because they will start at the cell surface and go down deep into the actual muscle. This is what is going to allow for communication that's on a cell surface. Think action potentials, cell signals. It'll take them and then dive deep down into the cell, carrying that signal to each individual myofibril within. And those myofibrils, as we mentioned, parallel lengths across the cell. So each one of these lines, you can see the green and blue lines coming out here, those are the myofibrils. Now, these myofibrils are really densely packed within the actual cell. You're going to end up with thousands of these per muscle fiber or per muscle cell. So each one is running in parallel to the others. So they're all one parallel the entire length of the cell. And it's these myofibrils that contain the contractile elements, the pieces that will actually contract and cause your muscles to shorten. Those are known as myofilaments. So the myofilaments are inside the myofibrils. The myofibrils are inside the muscle cell. So we have one cell, then all these parallel fibers, that's the myofibrils. Then inside the myofibrils, looking at these myofilaments. So here again, we've seen this picture before in a previous video, but just to rehash again, look at it. This is a single myofibril. You'll notice there can be dozens of those, if not potentially hundreds of those, inside one single muscle cell. Now, if we blow that up in the bottom here, that's one single myofibril. You can then see how many dots are inside here. Each one of those dots is going to be the contractile element, the myofilaments. So you can have hundreds, if not thousands of those per myofibril. So let's look at the two components. They're thin filaments and thick filaments. The thin filaments are primarily made up of actin subunits. Now, actin, each one of these green spheres is one actin molecule. So each single green sphere is an actin molecule. You'll notice these actin molecules are all tightly held to each other, forming this double spiraling strand. Almost basically a double helix there of actin moving around. The actin subunits are where you'll find the myosin binding site. We'll get to what that means in a second. But basically think about this. The actin is where the other part of it, the thick filament, can bind. So we have the actin subunits containing the myosin binding site. Tropomyosin. Tropomyosin is this orange line that's going across here. You'll notice that tropomyosin is blocking the binding site on actin. That means the thick filament can't bind if tropomyosin is in the way. So tropomyosin helps to keep the thick filament from binding when the muscle is relaxed. But when the muscles contract, we have to move that tropomyosin out of the way. In order to do that, that's where troponin comes into play. Troponin is going to be these small little units here. There's one, here's one, here's one. That's troponin. They're there as their sole purpose is to help and hold tropomyosin in place until it's time to move it. Then troponin helps to move tropomyosin out of the way. So troponin is controlling the placement of tropomyosin. So looking at the whole structure put together, here we have the double strand of actin, the thin filament. That is what each one of these green lines up here is showing. So each green line is one of these spirals of actin with tropomyosin with troponin. That's the thin filament. Now we have to build the other half. That's the thick filament. That's this other stuff, the purple stuff. The thick filaments are made of myosin. You'll see there are two myosin strands interwoven together. That's why you have two myosin heads here. The two tails are spun together. That's one unit. Two myosin molecules make one unit. Now we're going to have all those being woven together to create the thick filament. You'll see there's myosin heads sticking out at all different places. They're sticking out at a full 360 degrees around that strand. And they're staggered down the entire length. That's because these myosin heads need to attach to the actin units. That's why there's myosin binding sites. So if we take the two pieces here, we know the green lines are the actin. The purple lines are these thick, which is mostly myosin, thin and thick filaments. They'll overlap to create that sarcomere. So this is how we're building the functional unit inside of the muscle cells. So that's how we're building this sarcomere, as we mentioned. Now I've gone through this sarcomere a little bit in the last video, but let's kind of rehash and make sure we're on the same page. First of all, this sarcomere is going to be one region of the overall myofibril. So it's only one part. You'll have one sarcomere, a second sarcomere, a third sarcomere, a fourth sarcomere. They just run end to end down the entire myofibril. This is the smallest contractile unit. Because anything smaller than that, thick filament, thin filament, they can't do anything by themselves. This is the smallest unit, the sarcomere, that can actually contract and move. So Z-disc, we mentioned the Z-discs are going to be the borders, the ends of the sarcomere. So that is what is defining what each sarcomere is. I-bands we mentioned, so I-bands are these areas right here with only the thin filaments, so only the actin molecules. The I-bands do not have any thick filaments, so I-bands only thin filaments. A-bands, well, the A-bands are running the length of the actual thick filaments. So from the end of that thick filament to the end of the thick filament, that is one A-band. You'll notice there are regions that will be overlapping. So this area, for example, right here, is going to have overlapping thick and thin filaments. Still part of the A-band, but just that portion is an overlapping section. So these A-bands are what you see under the microscope as the dark striations. The light bands between the striations, those are the I-bands. At the very center of the A-band is going to be this H-zone. You'll notice the H-zone does not have any overlapping thick and thin filaments. The H-zone is only thick filaments, no thin. So that's the H-zone. So we start putting the pieces together here. You have your thick and thin filaments, so think actin and myosin, overlapping in the A band. That's the center of the sarcomere. The edges of the sarcomere are the I bands, going out to the Z disc. The I bands are only thin filaments. So the way it's built is this is your Z disc to Z disc, one sarcomere. You have an A band here. Here's a second A band on the sarcomere next to it. Here's another A band of the sarcomere on the other side. So we have all these repeating patterns, sarcomere after sarcomere. Now, when the thin filaments are at relaxed state, they're only overlapping a little bit. So thick and thin overlap a little bit. You have your M line, you're going to have your H zone. But now, once contraction starts, then the thick filaments, the myosin heads, attach and bind to the thin filament and pull it to the middle. Bind and pull, bind and pull. What they're doing is they're causing these thin filaments to move towards the middle, to move towards that middle line, that H zone, and basically fill up the H zone because there's no longer an H zone present there. So as the muscle contracts, those thin filaments are pulled towards the middle of the sarcomere until they touch. That's the maximum contraction for that sarcomere. Can't go any further. So what's happening here, you can see, each one of these little myosin heads can attach. As they attach, they move the thin filament towards the center. Eventually, as they do that several times, it reaches the center point, the M line here. Once it reaches that M line, the whole entire sarcomere has shortened. So in this case, sarcomere is shorter, the muscle has contracted. The I bands get thinner because you have this large area that's I band here, not as much smaller area, because the thin filaments are overlapping the thick filaments further. You're going to have the H zone disappearing. Thin filaments are going towards the center. Your Z line, the space between them, decreases because the sarcomere shortens. A bands, they don't change in size. They're still the same size. But each A band gets closer to the next one. So all the A bands get closer to each other. The sarcomere itself gets thinner. Contraction is occurring. And while all these sarcomeres each only move a little bit, but you add that up over several hundred, if not thousands, the whole entire muscle is now shortening. So your muscle itself is working because the thick and thin filaments end up overlapping, creating a shorter sarcomere, which creates a shorter overall muscle itself. The start of what muscle contraction is.